In this lecture, we're going to cover antibiotics and susceptibility. So the antibiotics that we use to target these drugs, this is what the doctors really want to know from us in microbiology. Historically, we're going to go through a little bit of history, but exactly how do these drugs work? What do they target in the bacteria? And then concurrently, what are the modes of susceptibility of resistance, pardon me, that these bugs develop because of that? Historically, obviously, humankind has dealt with infections of all sorts for, since there have been humans. So the mechanisms that we've had to treat them have really, even though there are lots of different ways historically in different cultures, if you really look closely at them, they all have a couple of things in common. And the biggest one, the biggest thing I should say that they have in common are the molds. Okay, if you look back as far as 1640, you can see that there are actually books written on pharmacology that recommended the use of molds. And if we look back even farther than that, we can find historical evidence that cultures used molds of some sort or material that might have had mold in it, frequently that would target an infected wound. So, for example, the Greeks and Indians, they use molds as well as plants, which the root of a plant is filled with mold, and they would use those as a treatment for an infection. In Greece and in Serbia, their recommendation was to cover the wound with moldy bread. So that worked. That could work in those areas. Warm soil, not easy to find in Russia, but warm soil was the treatment of choice for the peasantry in Russia. The Sumerian doctors, so go way back, Sumerian doctors, really fun. They would treat their patients with beer soup, and they'd mix in like turtle shells and stuff like that. But beer soup, the beer would have yeast in it. And then Babylonian doctors, there's evidence that they used things like sour milk, of course, that's soured, and then frog bile, which would have quite a lot to work against bacteria. And they would use that. Actually, there's evidence that they used it in infected eyes. So it's pretty, bold move, but it did work for some of their patients. And then if we go back and we look at some military documentation in the Sri Lankan army, they are actually instructing their soldiers. Obviously, soldiers have to deal with lots of wounds. So one of their instructions to their soldiers was to use this something called oil cake, which was apparently some kind of sweetmeat, like a minced meat, to serve both as a desiccant. If it's a sweetmeat, that's got a lot of salt in it. The salt will draw out the moisture, and also as an antibacterial. And certainly, fermented meats would have would have molds in them. So those are historically, you can see if you move through this chart as far back as 1640, but if you go through, 1870, they're still using mold. We got mold in 1871, mold in 1875, 1877, here comes Louis Pasteur, our hero, and he actually found it wasn't just mold that did that bacteria, in his case, anthrax bacteria, Louis Pasteur was nothing if not bold. Anthrax bacteria could actually target other bacteria. And we still, 1897, turn of the century, what are they using? Mold, okay? But what changes around this time between these years? What changes? Well, a lot of what changes is the technology. A lot of advances in the techniques in chemistry have changed so that, okay, about 1897, we use mold to get things done, but we can actually start looking at what part of the mold. We can extract chemicals and dilute things in using the technology of new chemistry technology, I should say, in order to do that. And Alexander Fleming was a doctor who was able to use those chemical techniques to really advance the world of pharmacology amazingly. Now, first of all, his major interest really came from the time when he was a military doctor. And Alexander Fleming, of course, military doctors lose a lot of patients, if not to their wounds, then next to the infections that follow. And it was Alexander Fleming's wish, or sort of his passion, to try and find some kind of antiseptic, some kind of naturally occurring sterilizer. And he was able to do that. He was able to extract the enzyme lysozyme. He did that in 1921, and he was able to use that successfully in surgeries. And then in 1928, as much as he really liked cleanliness in the surgical suite, he was a bit of a slob in his laboratory, and so I'm sure you've heard the story before. All his dishes were all sort of mixed together. His food dishes, his culture plates. So he had a culture plate of Penicillium notum, and a culture dish of staph. And what he noted, just sort of off the cuff, as he was observing, that the penicillin was able to not just inhibit but destroy the staph, and he was able to extract from the Penicillium notum what he called mold juice initially, but this secretion of the penicillin, and he found that even when he diluted that up to 800 times, he was still able to get bactericidal effects, where it was able to kill the bacteria to a certain extent. Okay, and so this was a big breakthrough, and it was after that you can see the progress continues. We have Prontosil in the 30s. We have streptomycin, chloramphenicol in the 40s and 50s, tetracycline. All of these were discovered fairly quickly. One of the things I want to point out is, what have we got going on here? We have the end of one war and leading into another war, which provides an awful lot of wounds to work on. And it was in 1942 that the term antibiotic was actually coined. Okay, so it's not that Alexander Fleming coined it; it was actually coined by Selman Waxman, but it's obviously an appropriate term. Okay, now as we move through this, it's very important to understand that there are two different types of antibiotics. Okay, we've got antibiotics which are bacteriostatic, and we've got antibiotics which are bactericidal. You should be able to tell from the name what the difference is, but basically a bactericidal antibiotic is able to actually kill the bacteria, usually by interfering with some major component of the bacterial metabolism. In many cases, the cell wall is a big one. Okay, whereas a bacteriostatic antibiotic actually doesn't outright kill the bacteria, but it interferes with its reproduction. It prevents it from multiplying. And one of the major ways, of course, DNA replication is inhibited, and other aspects of cell metabolism can be hindered, preventing the organism from multiplying, from thriving. That allows your immune system to catch up with the process of overcoming the infection. Okay, so not all antibiotics actually kill the organism. Some of them just hold them in abeyance so that your immune system can come into play. And I want you to know the examples. Okay, look at the examples here. Probably the most important part of any antibiotic choice that a doctor has to make: does that antibiotic actually function at the site of the infection? How can I get it to the site of the infection? So a lot of a lot of wounds, if you're talking about a wound infection. Most wound infections can be superficial. So in those cases, if you have a cream, you can directly apply the antibiotic to the wound. And we all have Neosporin or some other form of, triple antibiotic. Usually it's bacitracin, things like that in it. That we can smear on to a minor wound at home to prevent an infection or to overcome an infection. When you're talking about infections inside the body, serum, blood, CSF, urine, those kinds of infections, we have to make sure that we understand how to get the drug in a working, active form to the site of the infection. Okay, so we have to start with, well, how are we going to give it to the patient? There are 3 mechanisms of entry into the body for any antibiotic. Oral, take it through your mouth, intravenous, and through an IV, or intramuscular. Those 3 things become important. How am I going to get this to the site of the infection? Okay. In addition, it has to be able to get there, and this is where the intravenous and the intramuscular frequently come in to play. It has to actually reach the site in its active form. Okay, so if you give a drug, if you give not a drug, an antibiotic to a person, and it's degraded by stomach enzymes, it's never going to reach the site of the infection if the infection is a urinary tract infection. So all of these things have to be considered. Some drugs must be given intravenously or intramuscularly to avoid actually degradation by normal body responses. Antibiotics are not, they're not considered, they are considered foreign to your body. So your body's natural instinct would be, I'm going to get rid of this stuff. Okay, so we have to make sure that we overcome that for the body. Then we also have to make sure that the drug can reach the site in a useful concentration. Okay, and the best example of that really is nitrofurantoin. Nitrofurantoin, we can get it into the body, but it is eliminated so quickly by the body at the kidney that it really can't achieve any real concentrations anywhere else in the body except in the bladder, in the urinary tract. And so nitrofurantoin is almost exclusively, you can see here where it's used, it's almost exclusively used for urinary tract infections because it's eliminated too quickly to achieve a concentration elsewhere. So that's something that has to be, and we know the general trends for humans, but each person is different. So for example, here we're seeing clindamycin can achieve acceptable levels in the serum of blood. In most people, some people might not be able to. And so those are things that we do levels and things like that to check for. Then the drug has to be able to interfere with whatever the infecting agent is. So big important thing here, this is why the doctors want to know right away, is this a gram-negative or is this a gram-positive? The majority of our, or I shouldn't say the majority, many of our drugs target the cell wall. That's the difference between a gram-positive and gram-negative. You have to make sure that you're using the right drug for the right bug. Secondarily, penetration of the cell wall is important, and that'll be different if the organism is gram-positive or gram-negative. It's all important to every drug. So we need to make sure that we are using a gram-negative drug against a gram-negative bug, and so on. So what we're seeing here is you can have an active drug that you give to the patient. It has to go through several steps to make sure that it actually works. First of all, it has to actually reach the site of the infection. It has to be able to be absorbed and is functional against that particular organism. It has to be able to be taken up by that organism, either one of these, and it actually has to work against the target, the organism, leading to the either bactericidal effects or static effects. Okay, any one of these, if you have a problem with any one of these, we can end up with a failed antibiotic therapy. Okay. In addition, any one of these that might change through the course of the infection will lead to resistance of that bug. Okay. So some of the things that you should know from this certainly are what are the things that we're looking for efficacy, but also know the relative sites. How well do these drugs work at their various sites? Noting norfloxacin and nitrofurantoin, they're not intended for serum or CSF at all. Okay, gentamicin, clindamycin, not intended for CSF. Okay, the ones that you want to look for at CSF are ampicillin and ceftriaxone. CSF, you have to be able to, the drug has to be able to cross the blood-brain barrier. And what we're seeing here is that ampicillin can, gentamicin can't. Most drugs we can reach pretty good concentrations in the blood. The examples are those ones that are eliminated so quickly by the kidneys. Okay, now we're going to go through the modes of action. Okay, and we're going to start with, well, what are the targets? Organisms, of course, bacterial organisms have lots of targets that we would like to hit. It may seem, why don't we just hit any old target? But the idea here is to increase the effect on the bacteria and decrease the effect on the human. Okay, so let's target those things that only a bacteria has and that humans don't. Okay, and that way our human cells will be spared. Okay, that doesn't take into account allergies. Allergies is something you always have to worry about. Okay, and is completely separate from a mode of action. So obviously one of the major sites where we're going to look as a target is the cell wall, since none of our cells have a cell wall. Okay, so we really aren't targeting anything that's happening for us if we target cell wall synthesis. Okay, most bacteria, not all, but most bacteria do have a cell wall, and it's made of peptidoglycan, which will be continuously synthesized by the bacteria in order for it to survive. So just like your skin has to be synthesized continuously, it wears out. Same thing with the bacteria, just on a really tiny basis. Okay, so we can target cell wall synthesis, and you can see the drugs that do that. You have your beta-lactams, your penicillins, cephalosporins, carbapenems and monobactams, as well as vancomycin and bacitracin, and which target specifically the cell wall. We can also consider polymyxins. They don't really target the cell wall, they target the cell membrane. That's important because polymyxins will have higher toxicity rates than beta-lactams. Vanco is pretty toxic, and bacitracin, but for other reasons. Then we can also target nucleic acid synthesis. So you might be saying to yourself, wait a minute, we have nucleic acids. We synthesize nucleic acids. And that's true, we do. However, the enzymes and the cofactors that are used by the bacteria are slightly different. And so we can target those and have, you would have higher toxicity rates in humans with these drugs. But they do target, and they will help slow the rate of growth for any infection. Okay, and our major players there are those that target folate synthesis, which is a coenzyme required for nucleic acid synthesis, and that would be the sulfonamides and your trimethoprim. DNA gyrase, which is important for the unwinding of the DNA so that it can be expressed. The quinolones will target that. And RNA polymerase, of course, rifampin will target that. One of the other things to remember is that similar to the way we treat cancer cells, Bacteria have a much higher metabolism. They reproduce much more quickly than human cells. And so by giving you a dose of these antibiotics, the idea is, yes, we might have some toxicity to human cells, but because the bacterial cells are replicating more, which means they need to undergo nucleic acid synthesis more often, they will be the most highly targeted in the area. And then finally, of course, we have protein synthesis. And again, you might be saying, wait a minute, we synthesize proteins. But the difference, those subunits, really what we're targeting here are the ribosomal subunits. And our ribosomal subunits are not the same as bacterial. They are different. So we have a 70S, and we have, I think it's a 70 and a 40S ribosomal. Don't quote me on that. In a bacteria, it's 50S and 30S. And so we can target those subunits. There is no subunit in eukaryotic cells which will compare. And so these drugs do target specifically the organisms, the bacterial organisms. And you can see some target the large subunit, the 50S, and some target the small subunit, the 30S. The small ones are the tetracyclines and the aminoglycosides. The 50S, macrolides, Clinda, linezolid, chloramphenicol, and streptogramins. So all of these, look at the large choice of antibiotics that you have to choose from with different targets. So we're going to start with the beta-lactams because, of course, that's where it all started with with the first bacteria antibiotic, being isolated as penicillin. We know now what makes that important is the fact that it is a beta-lactam. It has a beta-lactam ring target that is able to be the functional part of these antibiotics. And what these are targeting specifically are the enzymes essential for peptidoglycan synthesis. And these enzymes are actually anchored in the cell membrane, and as a group, all of these enzymes together, they kind of hang out in a little group, are referred to as penicillin binding proteins, or PBPs. Okay, so these PBPs, I keep losing my pen. These PBPs, they kind of hang out in a group in the cell membrane so that they can continually reproduce peptidoglycan as it degrades over time. And there's lots of different species of penicillin binding proteins. And even one organism can contain 4 or 5 different types of PBPs, okay? Their major function is to actually create the cross-linking within the peptidoglycan. So technically, even though we call them penicillin binding proteins, their technical name would be a transpeptidase because they create that cross-link between the molecules in peptidoglycan. And they're critical for survival, obviously. The organism needs its cell wall for survival. So what happens is the penicillins, the beta-lactams, what they do is they bind to the PBPs, inactivating them. They're unable to function. And so cell wall synthesis is ended. It doesn't happen anymore. Okay? So and it's the beta-lactam ring that is the active component of each of these drugs. Okay? A lot of what changes when you go from one drug to the other, there's a lot of other outliers that actually serve to protect that beta-lactam ring, okay, from lysis by the organism. What generally happens when that beta-lactam ring binds to those PBPs and inactivates them, you have no more peptidoglycan being produced, and so osmotic forces that the cell wall helps to control are able to create instability, and the bacteria frequently lyses. So that's where it's a lytic, pardon me, a a bactericidal or a lytic event that frequently occurs. Okay. Um, pardon me. Unfortunately for all of us, as soon as we started using these drugs, the bacteria, both positive and negative, were able to start producing beta-lactamases that are able to cleave that beta-lactam ring, which makes it non-functional, which means that it doesn't bind to the penicillin binding protein, which means then the organism is able to survive. Okay. Now, they don't necessarily, the beta-lactamases that are produced by gram-positives are not necessarily produced in the same way as in gram-negatives. So remember, the gram-negative is kind of a layered cell wall, whereas the gram-positive is one big thick clump of cell wall. And the gram-positive cells, what they do is they create the beta-lactamase and excrete it into the environment. Okay, whereas gram-negative cells actually create the beta-lactamases, and they actually maintain those in the periplasmic space in that little layered cell wall, which means that you actually end up with increased resistance to beta-lactams for gram-negatives because the concentration of the beta-lactamases that they produce, and there are many different types of beta-lactamases, but the type of the concentration is much higher, and so they end up being much more resistant to these particular classes of drugs than gram positives. So even though it may not seem like it, many of these are considered only for gram positive organisms because gram negatives are pretty well resistant to them. Then we have a group of organisms called the glycoproteins. It's not really a very big group. It contains vancomycin and bacitracin. Okay. Now these actually they do work against the cell wall, but they don't actually bind to those penicillin binding proteins. Okay. So they are targeting the cell wall, but what they actually do is they interfere with the ability of the enzymes to incorporate precursors into the growing cell wall. And the major factor is, and I tried to show you this here with the chemical, look at the size of this sucker. Both of these, they're huge, okay? And that's basically what they do. They just get in the way. They just the enzymes are there, the enzymes are functional, but they can't actually reach. They never, remember what's the important thing about enzyme function? They actually have to be able to bring the reagents close together. These chemicals prevent that. They're enormous, okay? The size of these drugs is what makes their function work. However, the size of these drugs will also affect their function. So vancomycin, for example, which is the larger of the two, it's enormous. This will actually have problems with cell uptake in gram-negative. So as far as using vancomycin against gram-negatives, it's a little bit less successful for that reason. Okay, bacitracin, which does the same thing. It's just big and it gets in the way. It can inhibit a lot of metabolites which are required, using them over again. Same way, it just gets in the way. It's so big, okay? Here's the problem with these two. Both of them, bacitracin has a significant potential toxicity. So as a general rule, bacitracin is pretty much only used as a topical agent since we don't really want to eat bacitracin. It really is pretty toxic in general. Vancomycin also has increased toxicity specifically for the liver, and a lot of that is related to the enormity of the molecule. Remember, none of these are considered friendly to your body, okay? Even though they're meant to be there to do a good thing for you, your liver doesn't recognize that. They only recognize it as foreign. Get this thing out of here. I don't like it. unfortunately, it can have pretty high toxicity. So levels of vancomycin, when you're being treated with it, have to be closely monitored to make sure that the patient isn't being overcome by the drug secondary to its purpose. Okay, it just really wonks out your liver. Then we have cell membrane synthesis, another target. Now remember, you have cell membranes, so these will have a little bit higher level of regular toxicity, but again, increased metabolism of the bacterial cells mean that these should function much more quickly and have more effect on the more rapid or the higher metabolism cells, the bacterial cells. We've got several drugs, daptomycin, missed my pen, daptomycin, as well as the polymyxins, which include polymyxin B and colistin. Both of these target cell membrane synthesis. And basically, the purpose of them is that they create leaky membranes. Again, you're just trying to disrupt those osmotic controls that the bacteria should have over its cell, leading to lysis. Okay, daptomycin is actually a lipopeptide agent, and it actually binds to and disrupts the cell membrane, and that's what this lovely picture that the manufacturers of daptomycin provide for us here. You can see it can incorporate itself in, and it likes to form a group, with its other members in the cell membrane, which creates a channel, and you lose electrolytes. Osmotic forces, not so good. Okay, now these daptomycin in particular has pretty potent activity against gram-positive organisms, and including those that are resistant to other agents. So this is something that we use when we have a more resistant organism, in particular in the gram-positives, for a patient. It's a little bit higher human toxicity, so we would probably not use this as a first line of defense. Polymyxins were discovered, they're kind of old. We knew about those a while ago. They're not used all that often right now, again, because of their target cell membrane. And they are able to do that, disrupt cell membrane members, which will lead to leakage similar to daptomycin. Okay. The problem with these two is going to be that human toxicity. These are generally considered the agents of last resort because of the human toxicity. In general, they're used against gram-negative bacilli, things like Acinetobacter, things like Pseudomonas, which develop high levels of resistance, but they have not developed resistance to these. These again, a little bit more. We try other things first before we had to go on with these. Another target, protein synthesis. Remember, when we're talking about protein synthesis, we're talking about targeting those subunits. Okay, these antibiotic classes act by inhibiting the function of those subunits by binding to it or either binding to it directly or preventing those two subunits from coming together in a functional ribosome. Okay, each subunit separately, not very useful. Okay, these classes include aminoglycosides, macrolides, lincolnosides, streptogramins, which is called the MLS group, the ketolides, chloramphenicol, tetracycline, oxylidonines. They're going to come out with a whole bunch of new ones, I'm sure. There's lots of research in this area. And while they're generally all considered the same, they all inhibit protein synthesis. Their actual functions of these different groups are vastly different. Okay, so let's look at the different targets in protein synthesis that we have. Again, as I said, there are multiple targets in this process that we can these drugs can look at. The biggest thing is the fact that their subunits are different. So when the subunits are separated, and unfortunately this doesn't, well, it is here. So when the subunits are separated, if I can bind to these sites here, either one of these sites, then there's no way that any messenger RNA can allow the subunits to come together, meaning no proteins are produced. Some of the other things that can happen is the actual site where for elongation that can be blocked. So there's a lot of targets that can be prevented so that the proteins are not produced by the bacteria. So we'll go through each of the different groups, and just starting with the aminoglycosides, we deal with those a lot in the laboratory. Unfortunately, resistance to these is increasing over the more we use these drugs. Aminoglycosides bind to the 30S subunit, ribosomal subunit, which interrupts any kind of ribosomal formation or mRNA binding, okay? And which obviously prevents any protein. Just kind of know what the drug binds to and what it's preventing. So it binds to the 30S subunit, it's preventing protein synthesis. And what do we include in the aminoglycosides? So that's the class. What are the drugs? Tobramycin, that's not used that much, but amikacin is used all the time. Netilmycin, streptomycin for TB, very common. Kanamycin for TB, very common. Okay. The key thing here, no anaerobes. Anaerobic bacteria are not able to take up these agents intracellularly. They don't have the ability to take it. Remember, this drug has to reach the inside of the bacteria. Anaerobes cannot do that with aminoglycosides. So you would never use any of these drugs, which means you would never report any of these drugs on an anaerobe. Then we have our MLS group, which is our macrolides, our lincosamides, and streptogramins, okay? These bind the 50S subunit, and they include, which of course prevents the protein synthesis, and they include erythromycin, azithromycin, clarithromycin, clindamycin, and then streptogramin B, which is the only member of its group at this point. These drugs primarily are used for gram-positives. They might be reported, erythromycin, for example, might be reported clindamycin on a gram-negative here or there, but primarily these would be used for gram-positive organisms. So the key thing to know here is that if I'm asking you which of these drugs should you not report on a gram-negative, you can report some of these on gram-negatives, but their uptake in gram-negatives is not very good, so that's why we generally use them on gram-positives. However, the no anaerobes rule for aminoglycosides, this is hard and fast, okay? We don't cross this. So if you have, what's your anaerobes? You have to know that the organism is an anaerobe, and then know that you would not report these drugs on it. More targets for protein synthesis, or more classifications for protein synthesis. We have the ketolides, which include telithromycin. Again, that's not something that we've reported. It depends totally on your pharmacy, what's on their formulary. These bind also the 50S subunit. The only type, the drug is different from the MLS group, so it gets its new name. Although it is similar to the macrolides. This does still, this telithromycin, this does remain effective so far in gram-positives that are resistant to other macrolides. So this is an emerging one. It's relatively new, so it might be added to your formulary if a lot of resistance develops in your population. And one of the things that it does, and you can see here, the company gives us a lovely little picture. The reason it doesn't have that resistance yet is it doesn't induce the methylase, the MLS methylase that is produced by some bacteria resistant to macrolides. Then ocazolinids or linezolid is really the only one in this group. It's a synthetic agent and it inhibits protein synthesis actually through a pretty unique mechanism. And its mechanism is to prevent the coupling of the two subunits with the messenger RNA. Okay, so this is a unique method, so it's not, because it's a coupling factor that's prevented, it's not expected to be affected by the resistance mechanisms that are caused, that are present with other drugs. So we're hoping that linezolid lasts longer, if that's a good thing. Okay, but that's in many formularies at this point. And more targets for protein synthesis. We have lots and lots in this group. You have chloramphenicol. Chloramphenicol binds, again, the 50S subunit. And what it does, actually, when it binds, it prevents the, remember I said it's not, this one, remember, you prevent the elongation. That's what chloramphenicol does. It binds to the subunit and it prevents the formation of any peptide bond. You can kind of see that. If I can get my pen working, here in this part of the thing. So no peptide bond is formed. So it doesn't really, the ribosome could bind transfer RNA, but it can't add any amino acids to the next growing chain. So, well, that's a bummer. Protein synthesis is prevented. The good thing is this is going to be broad spectrum toxicity, broad spectrum use. Okay, it works against gram negatives, gram positives. However, there's an awful lot of toxicity associated with it, secondary. So your liver really doesn't like chloramphenicol. Okay, so for that reason, it's not widespread usage. The tetracyclines, on the other side, these inhibit protein synthesis by binding to the 30S subunit. So this is the small subunit that they're binding. And, pardon me, what they're preventing is incoming transfer RNAs. They can't actually read the codon, so they're unable, and you can see that here in this area, they're actually unable to bind. They do have to bind in order for any peptide to occur. So in the if there's any other antibiotic present, you might have multiple targets working at the same time. The tetracyclines do have a broad spectrum of activity. They work against gram negatives and gram positives, and they also do work against several intracellular bacteria such as chlamydia, rickettsia, things like that. So remember, those organisms are they don't have a cell wall that we can target. They're always inside the cell. So they have to be able to be taken up by the human cell and target the bacterial cell inside, which does work. This picture is pretty good in the sense that it does have, even though they're not in this class, I just wanted to show you, you can see where the erythromycin is working in the streptomycin, but those weren't germane to this particular slide. Then we have, finally, last group of protein synthetics are the glycogenes. I'm probably not saying that right. These are synthetic derivatives. They are related to tetracycline. You could see the only member in this is tigecycline. And it's the first member of this group to be approved for clinical use. So it's pretty new. And what they do is they bind to the 30S subunit. Okay, so again, this is like tetracycline, small subunit. But what's the difference between this and tetracycline? Tigecycline is refractory. So what that means is that lots of gram negatives and gram positives that have developed resistance mechanisms to tetracycline still exhibit sensitivity to tigecycline. So that is refractory, so it's still working, and it doesn't seem to be able to, the resistance mechanisms don't work for that at this point. So it is a new, relatively new molecule to add to our formularies. Another target would be our nucleic acid synthesis. Remember, what are we going for here? We're going for the fact that the increased metabolism of the bacterial cells will mean that they will be an increased target, a higher target than our cells. Okay, so we have several groups. We have the fluoroquinolones here. Okay, frequently we just call them the quinolones. And these are actually what they are derivatives of naldixic acid, which is an older antibacterial agent. We don't really use that anymore except in our lab. And the examples are ciprofloxacin and ofloxacin. These are two examples of, pardon me, of quinolones. And what they do is they bind to and interfere with DNA gyrase enzymes, which would be involved in DNA supercoiling, which is essential. So they actually help to uncoil the bacterial genome, or any genome actually, but the bacterial genome, to allow for transcription, replication, those things. So, pardon me. The DNA gyrase enzymes, when they're interfered with, neither replication nor transcription can take place. So that's pretty direct interference with metabolism of the cell. And the quinolones at this point are fairly broad spectrum. Then we have metronidazole. Metronidazole is exclusively used for anaerobes. Okay, that's important to remember. We're not actually completely positive, or at least I'm not. I think they're still working on it, of the exact mechanism of how the molecule interferes with the anaerobic bacteria, but it does seem to create breaks in the DNA strands, which would of course interfere. I'm not sure how the breaks occur, but it does interfere with both replication and transcription. And the key thing here, and why it's used only in anaerobes, is that activation of metronidazole, or remember, you have to get it there in an active form. It is only active under conditions of low redox potential, which are found in anaerobic environments. So you really only use this in a situation where anaerobic bacteria are most likely. So, pardon me, abscesses, things like that. Now, and of course, most of those are gram-negative. So this does work best against anaerobic gram-negative organisms. Now, this potentially could work against, say, for example, a facultative anaerobe in an anaerobic environment, because that bug can grow there. But you would never use it, for example, against something like Pseudomonas, which is a strict aerobe, so it would not be growing in a condition of low redox potential. Please note the thought process that has to be there. Okay, level 3 question. So if you're asked, can you use metronidazole against a gram-negative like Pseudomonas aeruginosa, for example? Well, Pseudomonas aeruginosa is a strict aerobe. A strict aerobe would not be growing in the low redox potential of an anaerobic environment, which means metronidazole would not be active, which means it wouldn't work. However, if you were to say, could it work against E. coli in an abscess? Well, an abscess would still be a low redox potential anaerobic environment. Even though E. coli can grow aerobically, it is also a facultative anaerobe. So you have to think through the entire process when you answer questions like this. And then we have nucleic acid synthesis, the last group in this, well, it's not the last group completely, but rifampin is another group that directly binds with an enzyme involved in nucleic acid synthesis, and that's the RNA polymerase. It binds to the DNA-dependent RNA polymerase and inhibits the synthesis of any RNA. Now here's the deal. It's only used, rifampin, I should say, is only used against gram-positives, and that's because it cannot penetrate the outer membrane of the gram-negatives effectively. So as a general rule, it's not considered a gram-negative organism. The other problem is rifampin develops resistance very quickly, so it really should not be used on its own. It should be used in conjunction with other drugs. Okay, the folic acid pathway. This is important because folic acid is used by bacteria to produce many precursors that are necessary for DNA synthesis. So any drug that targets the folic acid pathway is considered to target nucleic acid synthesis, technically, even though you're kind of a step before that. Okay, so you can see in this example here, you have the different enzymes that are targeted by these folic acid I guess, triggers. You have the sulfonamides, which target the an early precursor enzyme, and you have trimethoprim, which targets a later precursor enzyme. Okay, and you can see what they're producing is still a precursor, but it leads to the production of thiamine, guanine, adenine, nucleotides, obviously necessary. Okay. The sulfonamides specifically, the enzyme that they're targeting is dihydropteroate synthetase. Okay, again, early in the folic acid pathway. There are several different sulfonamides that you can use, and they are broad spectrum, but they do not work against Pseudomonas aeruginosa. Okay, just that's the one, just don't use it against that. Remember, always remember your exceptions. They end up on tests. Okay. Trimethoprim, same pathways targeted, different enzymes. So dihydrofolate reductase is what's targeted. That's the specific enzyme, and that's inhibited. Its function is drastically inhibited by trimethoprims. Frequently, trimethoprims are combined with a sulfonamide. Sulfamethazole is one of them, to produce a single single drug. So you'll frequently hear doctors ask about trimethoprim sulfa, and that's what that is trimethoprim combined with a sulfonamide in order, it's a single formulation, but the drug is actually targeting both enzymes at the same time. Now, just to add to this list, I didn't write it on here, but the never mind. Okay, resistance mechanisms. Resistance mechanisms, what we have to understand is that there are three different types of resistance. You have intrinsic resistance, biologic resistance, and clinical resistance. And it's important to understand the difference between these. And we've kind of already talked about some, so it is important. Intrinsic resistance, for example, is important for you to understand because you would never report those drugs. Intrinsic resistance is when an antibiotic never would have worked against that bug for whatever reason. We just talked about it. Metronidazole, Pseudomonas aeruginosa, you would never combine those two. That's intrinsic resistance, okay? Biologic resistance is when you see decrease, you in the lab or whatever, are noticing changes in the bug, not in the drug, but in the bug, that are resulting in decreased sensitivity to a particular drug, okay? So aminoglycosides, I talked about that. We're seeing we need higher and higher dosages of aminoglycosides to work against bugs like Enterococcus, which are developing these high-level resistances to that particular drug. And then clinical resistance is when the sensitivity has decreased to such a point where it is no longer clinically useful to use that particular drug against that particular bug. Resistance must always be reported as a drug-bug combination. So you would consider, for example, in the example we just gave, Pseudomonas aeruginosa is intrinsically resistant to metronidazole. A bug in and of itself is not resistant alone. You have to have the drugs against which it is resistant. Okay. Some examples, you can see the activity of erythromycin, various aminoglycosides. We can see biologic resistance when we have any kind of decreasing pH, whereas the activity of tetracycline decreases with increasing pH. That's important. Where is your infection? What is the pH level at that site? And we have to take into account, okay, at this site, tetracycline will be less or more resistant because of the pH level. Aminoglycosides, for example, because their uptake is an oxidative process, you don't use aminoglycosides against anaerobes. We've already said that. You have to have that oxidative process present. Without oxygen, aminoglycosides are not an option. Okay, biologic resistance. Aminoglycosides can also be affected by the concentration of electrolytes, cations in particular, like calcium and magnesium. And that effect will be highly notable with things like Pseudomonas aeruginosa. Okay, and then thymidine concentrations will also affect the function of various antibiotics. So those are all things that you would have to take into account. Where is the infection, and how will that play with my resulting sensitivity level? Okay, so this chart is giving you a list of the intrinsic resistance that you should be aware of. I'm not going to read it to you. I want you to go through and understand natural or intrinsic resistance that's present. You would not report these drugs with these bugs. And just to go over the infection that you see, if you do a culture on an infection site on day 1 and you see a sensitivity pattern, over time and treatment of that infection, you'll see that you can see, especially if it's unsuccessful treatment, you can see that sensitivity pattern change, okay? And that is because of the way that bacteria grow. So we know that if a bacteria is sensitive to a particular drug, it'll die. That's the point, right? If it's resistant, it won't just die, but can also thrive. It is possible for them to thrive if you give them antibiotics, which will tend to reduce other bacteria in the area. So what happens with drug resistance? One of the things that you have to remember is in a particular colony, not all organisms are genetically identical. You do have outliers. You have ones that are genetically different and variation occurs. So if you have this colony of organisms and you treat with the bacteria with some kind of antibiotic that favors the survival of certain members of the colony, those organisms, because now they don't have to compete with the rest of the colony, those organisms will thrive. Okay, that's important to remember. So we have to make sure that we are taking into account all of the members of the colony. You never want to do your sensitivity from just one colony. You want to make sure you pick up more than one colony off the plate so that you take into account as many of the variations that exist as possible. The other thing to remember, those crazy bugs. Remember we said that bugs variation when you're talking about asexual organisms is limited to mutations, but we know that that's not true. Bacteria can transfer genetic material, and you can see here that copies of resistance genes can move from one organism to the other within a species and between species. which is our biggest, it's the bane of the microlab. So gene transfer allows, even if it's a different species, the production of highly resistant colonies in our patients. When you have a patient who's highly susceptible and you really want to get rid of that bug, you need to use a combination of drugs to be able to ensure that you're not just targeting this group here, but you're also targeting the really resistant group. And so who are the usual suspects for our resistant? Well, you should know some of them, and they include guys like MRSA, methicillin-resistant Staph aureus, VRE, or vancomycin-resistant Enterobacteriaceae, Enterococcus, vancomycin-resistant Enterococcus, ESBL, or extended spectrum beta-lactamase producers, CRES, carbapenem-resistant Enterobacteriaceae species, pan resistance, which is a term we use for those organisms which are demonstrating resistance to all of the drugs in our formulary, or nearly all of the drugs in our formulary. And then the thing that we're really nervous about, and we don't want it to emerge, we've only had a handful of cases in the United States, is VRSA, which is vancomycin-resistant Staph aureus. This can occur quite easily because Enterococcus is quite promiscuous. This is something you should know, and that it can transfer its resistance plasmid really across species. When you have a commingled infection between VRE and MRSA, we're very afraid that the Enterococcus will give its vancomycin resistance plasma to the MRSA and create the VRSA, which we're very afraid of because our treatments for that are very limited. And this is just a chart to show you how the resistance has increased over time. Look at the numbers. And there are a couple of reasons for this. There are a number of reasons for this. Number one has to be over-prescription or misprescription of drugs, okay? Overusing antibiotics when they're not needed. You'll note that none of these are going down, okay? They are all higher than when they started, okay? And that trend continues. They're all higher than when they are started. The other thing is we have a lot more immunocompromised patients who carry these suckers around. So getting rid of them is going to be, and always will be, very difficult. Detecting them is our job. So we do have routine screens for MRSA and sometimes VRE, depending on the population. The mechanisms for resistance, there are several, okay? For MRSA, our general thing, while we call it methicillin-resistant Staph aureus, most hospitals don't use methicillin. We actually use oxacillin, but nobody really wants to say ORSA. It doesn't really roll off the tongue very well, so we say MRSA. But it is a class of antibiotics, okay? And our major screening drug for MRSA is cefotaxime. Cefotaxime resistance, you can throw a cefotaxime disc down, and if the Staph aureus is resistant to that, it is an MRSA. Okay, that's our major easy marker. The other thing that we use is we use this MRSA plate, which is a chrome agar, and it contains oxacillin, actually is what it contains, but it contains 6 micrograms per milliliter. And if you can get growth of several organisms on that, but if it grows on this and it is mauve in color, mauve, not pink, mauve, then it is likely it is an MRSA. You just have to do the catalase and the coagulase to prove that it's actually a Staph aureus. There are other things that can grow in this. You know, Enterococcus can grow on it, yeast can grow on it, even other staphs, but they won't be mauve. Okay. The most common mechanism for resistance for MRSA is the MEK-A gene. And so the other thing we can do is we can actually do PCR to detect the MEK-A gene. The problem with using PCR as your method of detection is that you won't catch that subpopulation of organisms that are resistant by the MEC-C gene or other mechanisms. There are several different mechanisms, and you're looking at several mechanisms here in the one. I'm sorry, got my pen working again. You have 1, 2, 3, and 4. The things that are happening here. You wouldn't catch necessarily all of them. Although MECA is the most common, okay, MECA encodes a penicillin binding protein that it doesn't bind the methicillin or other beta-lactam antibiotics. So it's one of those things that enables the many bugs to survive, but it is not the only mechanism of methicillin resistance, okay? Sometimes you only discover the methicillin resistance when you run the panel and you see, well, wait a minute, I didn't have any signs that this was methicillin resistant, but look, it's not dying. It's growing in all of my wells. It's methicillin resistant by a different mechanism, okay? Now, resistance to via vancomycin, here we go. For vancomycin resistance, again, there are several mechanisms for organisms to be resistant to it. We usually, if your hospital screens for vancomycin resistance, they might do it with, this is a VRE chrome agar. There's also a VRE Mueller-Hinton's. If it grows on the Mueller-Hinton, there's something wrong. Nothing should really grow there. For the chrome agar, you can see here you have 2 different colors. Kind of hard to see here. You have like a teal color and then like a berry color here, like a kind of a purpley color there. And the difference there is that what you're finding here are the 2 most common kinds of vancomycin-resistant organisms, Faecium, which is purple, and the teal or the blue is Faecalis. Enterococcus faecium will show up purple on the chrome agar plate, or that berry color, and the Faecalis is blue, okay? Again, there are other mechanisms. You can, drop a vancodisc. There's a lot of the other mechanisms. If your population warrants screening regularly for this, you might screen for MRSA upon admission. You want to make sure that the patient had it when they came in, because then it's not your problem if they get it, okay? Insurance companies are starting to say we're not paying for nosocomial infections. So, but if they already had it when they came in, it's not your fault. They also will screen as when patients move floors so that you know exactly where they acquired the MRSA or the VRE, as it were. ESBLs, these are basically sort of heavy-duty beta-lactamases. They're able to hydrolyze the extended spectrum cephalosporins that have that really long side chain. So they're able to get to the beta-lactamase even through the side chain. That's why they're called extended spectrum. And the cephalosporins that are targeted include cefotaxime, ceftriaxone, cefotaxime, cefotaxidime, and aztreonam. Those are common ones that are targeted. And so we can use ESBL tests, manual tests such as this, where we test for things like we add cefotaxime or ceftazine. We usually do 2 drugs, and we include a beta-lactamase inhibitor. So you have the drug, you have the drug by itself, and you have the drug with some kind of beta-lactamase inhibitor, and you can see a drastic difference in the way that the pattern of the growth. I think if there's a difference of 3, it depends on the drug itself. I think in ceftazidime, it's 2 millimeters, and in ceftazidime, it's, I think it's ceftriaxone, cefotaxime, it's 3, and ceftazidime, it's 2 millimeters. But if there is a difference, then you have an ESBL. But you also have chrome agars. What's the manual methods are cheaper. That's the reality. Although it does cost, it is harder to do all of the, you have more, QC plates. So that's something that if this is a problem, your hospital might need to check for it. Just to show you for the ESBLs, this is the same plate, but what happens with the chromogers is that the organisms will grow up different colors. So you know that if it grows on this, it does have an ESBL, but what chromoger adds is a way to identify it as well. So the pink here, the growth of the pink one, is E. coli, and the blue one here is Kleb pneumo. That you can also grow a yellow one, which I don't have a picture of, which is Proteus mirabilis. Meaning you could grow them all three on the same plate. If all three grow, they'd grow different colors. Okay, so this allows you not only to identify the fact that it's an ESBL producer, but also to identify its actual organism ID. Okay, carbapenemases. Carbapenems are usually have been considered stable, famously stable, and they usually we had no problem with extended spectrum beta-lactamases and routine beta-lactamases. Carbapenemases unfortunately developed. These are a diverse group of beta-lactamases that are active not only against all of those cephalosporins that I just talked about, as well as cephamycins, but also against the carbapenems. So if you have a carbapenemase, you already know that you have an extended spectrum beta-lactamase. So it is an extended spectrum beta-lactamase on steroids, okay? The mechanisms of the interactions, you don't really need to know that. The key thing is that those big long side chains, they're useless. They're not protecting our beta-lactam ring, okay? I mean, these are just the mechanisms thrown out. I don't want you to get too caught up in it, okay? But basically, when we're looking for identification of these carbapenem resistant organisms, the drugs that we're looking for that used to be fine, they'd be sensitive, but now they're resistant are the imipenems, the ertapenems, and the meropenems. So when you're looking at your microtiter well, when you see those as resistant, in particular in your gram negatives, because those are the drugs that we use, you have a problem. And this is the major problem that we have in hospitals and nursing homes in particular across the country. Basically, a carbapenemase is just a really fancy, really powerful beta-lactamase. So finally, we're looking at a super chart here, which gives you the group of drugs, the type of drugs that are in that. So examples within that group, its action. Is it static or is it cidal? Side effects I don't need you to worry with too much. However, I do want you to know the uses. So note, okay, so the sulfonamides, they're not used too often because of resistance. You know, what are the uses? Who do we use it against? And then spectrum. So what's the spectrum on this? Let me find my pen here. Is it broad spectrum, or are we talking about, you know, where is it effective here? If it's broad spectrum, you can see it usually works against both gram positives and gram negatives. If it's not, it'll usually be restricted to one or the other, and so on down the line. So definitely be able to answer those questions. This chart will be very helpful.